Okay, welcome back, everyone. I hope you enjoyed the break and are being able to catch up with lots of your colleagues. We'll have a lot more time over lunch for that, which will be great. So now we're going to switch gears a little bit and talk about artificial intelligence and machine learning, which is actually a very important part of science and medicine today, because all of the sequencers, all of the synthesizers for DNA, they throw off lots and lots of data that need to be analyzed, and patterns need to be found in the data. And once those patterns are found, you have algorithms which can basically create ways to intervene, to cure diseases and do all kinds of other things. So right now we have two companies that are working in the area of artificial intelligence and machine learning. Very excited about this. So just briefly introduce both of yourselves and then we can jump in.
I'm Carlo Yuvienco. I'm Senior Director of Strategic Partnerships for an early stage biotechnology company called Envisagenics based in New York City.
Great.
I am Jacques Banchereau. I am an immunologist, and I'm the Chief Scientific Officer at Immunai, and delighted to be here.
I met Jacques, I don't know, 15 years ago when we were starting the New York Genome Center.
That's correct. I was the CSO at Hoffmann La Roche in Notley then.
That's right. And we did the opening ceremony together, the opening meeting together, kickoff launch, right?
That's correct.
And Carlo and I have been working together. He's been at the EDC for years and years, so also a great colleague. So Jacques, why don't you start off and let us know what you're doing at Immunai?
Okay, I have a few slides. I'm going to skip some probably, but Immunai is a 3.5 years old company that was founded by Noam Salomon, and who is our CEO. I joined as a CSO about 15 months ago. We have raised nearly $300 million, which is not bad for a startup. And have a group of about 140-something, 45, I think, people. We are headquartered in New York and stay in Alexandria, Center for Life Science, near NYU. But we have offices in Tel Aviv, as well as a lab in Tel Aviv, and offices in Zurich and Prague. Actually, Tel Aviv, Zurich, and Prague have a lot of our AI and ML colleagues. We have here in New York AI and ML colleagues. Next slide, please. And this shows you what we have done, the growth of the company in New York. 2 years ago, we were 3 employees, or they were 3 employees, in 360 square feet. They went to the biolab in NYU Langone, reached 33, probably was 25th or 26th, and then we are now 56 in 12,000 square feet. We are a little tight, but that permits us to interact actively. As you see, science in blue represent about 60% of the people here in New York, while ML and AI represent about 20%. The next slide shows you a little bit what we are trying to do. The immune system is immensely complex. And I became an immunologist because I hesitated between the neurobiology and immunology. Those are the two sciences where memory comes. You can remember this wonderful trip you had, or your body remembers that it has seen COVID. And eventually you have a less dramatic response when you see the bug again. And this is very complex. And because of that complexity, we have actually a lot of diseases that are associated to the dysfunctioning of the immune system. You see here in the center the normal state. Probably the main function of our immune system is to protect us against microbe, but it also has a role to eliminate all your dead cells. You make every day billions of dead cells, and they don't accumulate. So in other words, the immune system may be the garbage man of our body. But it's also a surveillance patrol against your cancer cells. It eliminates your cancer cells. So you understand that there is a problem if your immune system doesn't work too well. You can have infectious disease, you can have cancer. However, you know the other side. If it works too well, either you have autoimmunity or you have allergy. Actually, there are two fundamental forces in the immune system. You have a force which is the induction of tolerance, and another force which is the induction of immunity. Tolerance is amazing. When a woman starts having a baby, she has to tolerize the genes of the father. There's an enormous power that we need to understand that, and we don't understand it yet. So this balance, next slide please, is borne by cells and molecule. On the right panel, you see a slightly more complex version of what the doctor prescribes to you. When he says your differential counts in your blood, you see lymphocytes and you see monocytes and you may see neutrophil. The right panel here shows you that now with the novel tools, we can distinguish hundreds of population. And the distinction of this hundred of population is fundamental and totally permits us to understand diseases. So now we start seeing cells, but we also see molecule. On the left panel, I've described to you that there is three fundamental signals in the immune system globally. You have a signal 1, that is a specificity that kicks a bit the cells of the immune system. You have signal 2, which is modulation that can co-stimulate, help the signal, or co-inhibit, prevent the signal. And you have a signal 3, which I like to call the overdrive. Now, your signal 2 is basically like your brake and your gas pedal, except that the immune system has 7 or 8 different brakes and 7 or 8 different gas pedals. And this is what we have got to understand when you release the brake and when you need to push the gas pedal. And this is the whole thing. You have heard about the progresses of immuno-oncology, particularly with those molecules called checkpoint inhibitor. Those are molecules that prevents the co-inhibition to happen, because in the cancer, there's a lot of tolerance, there's a lot of inhibition. You want to block that. But this PD-1 targeting has been an extraordinary revolution. We have a hard time finding others. But we know there are others. And the whole problem is they act all in combination, and we need to understand that. Next slide, please. Next slide. So that's where, backward. Okay, no.
Sorry.
That's where AI and ML is. It's just like in the center, and is how much I understand of AI and ML. I understand maybe a little bit more than this. But what AI and ML does is take all the information that you have on the left side. If you do, for instance, look at the input. Those are everything that we are doing in the lab. We are getting samples from patients. We are analyzing by single-cell technology, by omics, which is all kinds of proteomics, lipidomics, glycomics, and so forth. We are doing experiments, functional genomics. We eliminate genes from cells. We add genes into cells. We do other experimental immunology. All that gets into our database that we call AMICA. And this is now that this whole set of data, together with data that are known. Artificial intelligence takes advantage of natural intelligence. All what has been created in the past 50 years in immunology is also integrated. That's why it requires extremely performing teams to put all that together. And now they are crunching the data. And they are crunching the data, and then they turn, and something comes. Now you need to say, oh, that's interesting. I have IL-92. Doesn't exist, but it's an example. IL-92 shows up, and you want to know, well, is that really important in that disease? Now you go back to the board and you do more experiments, functional genomics and experimental immunology. And basically the output is on the right. What is it that you find? You find novel target for intervening. You find predictive biomarkers. That means you see when you study patients, you see that some patients respond to your therapy and others do not. And now you can select the patients that respond. This is fundamental in the field of pharmacy. The big problem is there is this heterogeneity. And if you can select the responder patient, you're going to have many more drugs that are going to be utilized. It permits to optimize therapeutic combination. It permits to study enhanced preclinical model. And I'm going to go to 2 next slides, probably. Next. In other words, it is acting at every stage. We are using these approaches at every stage of the drug discovery. From discovering what is wrong in a cohort by this integrative analysis that permits us to generate lead molecule that we will do preclinical studies. When we see that we are on the right track, we can optimize the drug. And then probably where the importance is the highest is in the clinical testing. Again, understanding patients. And with that, I will just send you to the last slide where I have we are partnering. We are partnering with academia, we are partnering with biotech, we are partnering with big pharma, and to get to all those prioritized therapeutic indications, those optimized therapeutic combination. This is our blood, and one day we probably will also be generating our own drugs, which will be the best demonstration of the quality of what we are doing. The next slide is the final. It's thank you, and if you want more info, that's easy, info@immunai.com or Jacques@immunai.com. Thank you very much. Happy to address any question you might have.
Are there any questions? You're at the Alexandria Center, right?
We are there on the 11th floor. Mm-hmm. We are in not the building which is right near the water, it's the building before. We are above NYU.
At the rate you're growing, you're going to need new space pretty soon.
Well, we have been looking into that recently, but we have decided that it's a costly thing. We have looked particularly on the other side of the river. We have decided to squeeze ourselves a little bit, go back not as in the real days of cholera, I meant COVID, but that we're going to be working a little more from home, so we're going to put more space, more lab space, and because we want to really, in 2023, make the demonstration that the concept of the company is what the pharma of the future will be. So we don't want to be distracted in moving at this current stage. It's just a critical time where the inflection point is sometimes during the year.
Terrific. Anybody have a question? Yes, Brian.
What role, if any, will quantum computing, you think, play in drug discovery?
Well, I mean, it's not yet there, but it's obviously what we need. We are working with, for me, it's petabyte, but it's bigger than this. The amount of data that we are channeling and analyzing is gigantic. And we are I just hate to pose a question and have the answer 3 weeks later. I'm usually the kind of guy I say, hey, what if this is like this? And I like to have the answer in a minute, not 3 weeks later, because I forgot 3 weeks later what was my question. So better computers are definitely going to be fundamental. And of course, all the ML guys and all the AI guys develop better algorithms. They develop better softwares, better visualization, because eventually at some stage, the human intelligence will always have something to do. So we need to be able to see what the data integration will give us.
Okay, Carlo, tell us about Envisagenics, sorry.
Sure. So you can go to the next slide. The contact information was on the first slide for those who are interested. Actually, I'm substituting for my colleague who leads our data science team, Melissa Franck. She sends her regards. She wasn't able to make it today. But you have me, which is fortuitous because it's a bit of a homecoming, I feel, because I know a lot of the faces in the audience. And being at the EDC for 6 years prior to Envisagenics, it's probably more my field, this audience, than what I'm doing now, actually, for Envisagenics. But to focus back in on the company, so the company was a spin out from Cold Spring Harbor Laboratory in 2014. It's had, very, I would say, gradual but healthy growth over the years. It's a quintessential New York grit story, if you will, and that's one of the things that attracted me to join the team earlier this year. So the company spun out of Cold Spring Harbor, out of Adrian Krainer's lab, Martin Ackerman, where he was one of the only bioinformaticians working with the renowned biologists coming out and working with Adrian Krainer on RNA splicing biology. For those who are not familiar, so Adrian Krainer is one of the lead scientists responsible for developing the drug Spinraza, or known as nusinersen as well, which is effectively it's a cure for spinal muscular dystrophy, or atrophy, sorry, SMA, which at that point in time in 2016 was the leading cause of death among infants for genetically inherited diseases. So taking that inspiration, Martin founded and formed this company, Envisagenics, with Maria Luisa Pineda, all based on the premise that alternative splicing, which is a phenomenon in biology that is one of the root causes of SMA, but also associated with a plethora of other diseases, can be something to address with computational methods. And so that's where the AI and the machine learning comes into play from Martin's background. The company, and since we're a little bit more focused on where we were in terms of location, so the company grew out of Long Island. The company, from what I recollect, was placed in a launch pad somewhere in Huntington, and then quickly moved out of that to Grand Central Tech. So those of who are familiar, is Grand Central Tech is a co-working space located, now known as Company, in just off of Grand Central Station. And they were one of 1,000 that got accepted into that program, and that was their first incubator, being sort of this hybrid of a tech company and a life sciences company. They were there for a number of years, received a lot of notoriety and funding from folks like Peter Thiel's Launch Labs, Peter Thiel's, the name's Founders Fund, maybe. But then after that, graduated from there to J Labs. And then I would say that coincided with J Labs' founding in New York City. Was a very, very successful graduation into another incubator, now building out their wet lab team. And that also led to the partnerships with J&J. I would say that the network at J Labs was very fruitful for the company, and then later on introductions into Biogen, which was announced in the last 2 years. So we do have biopharma partners along for the ride with us. As I mentioned earlier, so we're focused on a number of therapeutic areas, primarily in oncology, but also alternative splicing, which I'll get into in a little bit more detail, is also known to be associated with some neurodegenerative diseases as well as metabolic diseases. We've got a proprietary drug platform that utilizes ML to not just uncover the RNA biology that is at work underlying some of these diseases, but also to provide prescriptions and guidance on developing drugs and therapeutics to counteract these diseases. You can go to the next slide, please. And so this is our pipeline of our core platform called Splice Core. We take in-house data as well as public data sets, as well as partnered data sets, all around RNA sequencing. Like I said, this is all based on RNA biology. And what we're trying to look for, essentially, is, if I could borrow a metaphor from the building industry, imagine RNA as a blueprint for constructing a house, and that house being protein in this sort of metaphor. And so protein is what allows our body to function, and that blueprint is the RNA that your body ends up sort of reading and then turning into a protein. But imagine, for instance, that someone comes along and you've got a stack of blueprints for your house, and somebody removes one of the sheets from that stack of blueprints, right? In RNA biology parlance, we would call that an alternative splicing event, where somebody comes in and excises out something that's critical to the structure of your house or your protein. Now, if you look at your house, right, after it's built, you would say it looks fairly complete, but then when you start to look at the functioning, you start to notice that it's missing a kitchen, right? So effectively, alternative splicing is trying to uncover some of those defects in the construction of proteins in our body, and a lot of those defects are associated with diseases. The machine learning piece of it is uncovering, well, how did that happen? You know, what was the cause of that alternative splicing event, and how might we now drug that event? And then on the output side, what pops out from our pipeline, in a sense, is different types of modalities of therapeutics, prime of which is antisense oligonucleotides, which is a type of RNA-based therapeutic. You can go to the next slide, please. And so the real value of this, and sort of a bridge to where this really hits the road, the rubber hits the road, is the amount of time that the in silico process takes, and the removal of a bottleneck of drug design. We can see that in 8 months' time, you can go from just taking in RNA sequencing data to popping out candidates ready for in vitro clinical testing. Next slide, please. And like I said, so we recently submitted a paper to bioRxiv to highlight the strengths of this platform. I encourage you to look it up. It's associated with the company. It was earlier this summer. And it was effectively is a case study in triple-negative breast cancer, and a proof of concept paper of this platform that we've developed. You can see here that alternative splicing, in terms of skipping on the left-hand side versus inclusion, you can see that the skipping of this, let's call it this part of the protein, is very strongly correlated with triple-negative breast cancer. And that correlation, sure enough, is also associated with a significant decrease in survival. Next slide, please. And so when we uncover this, like I said, the machine learning aspect comes into play of designing a drug now against that target, against that event of skipping that essential part of the protein. And so when we have developed an intervention, again, through machine learning, to now insert that missing component of the protein, we see a reduced survival of the breast cancer. And so now the breast cancer is no longer able to proliferate. Which is the takeaway from this slide. Next slide, please. And so as a company, our most advanced programs are in breast cancer in the RNA therapeutic space. We also have something that's trailing it as an IO therapeutic, and other oncology indications are also of interest to the company, and then probably least mature of which are also metabolic and neurodegenerative programs. ALS and Parkinson's are of interest to the company now. But as you can imagine, the challenge for us as an RNA biology company is the ingestion and acquisition of RNA sequencing data. And it has to be of a particular quality of data, which also makes it even more challenging, which is one of the issues that the company faces today.
Great. So you are located in Long Island City.
We are. So we're located in Long Island City in a science hotel, combined suite, 5,000 square feet in Long Island City, Alexandria's Bindery building.
Great. When did you move there? How is it?
This summer, and then we officially closed our doors in J Labs, at the beginning of the summer. It's been great so far. The team really loves it. I would say that the onset of COVID and sort of the company growing during the pandemic has made, particularly our software team, our data science team, and our bioinformatics team realize that they could manage things remotely. So if anything, there's more flex and hot desk space for that portion of staff, and a lot of the office is more dedicated to wet lab operations. So, I imagine, we probably would have been bigger if the necessity to have everyone in the same space, but we're trying to be efficient, similar to our colleagues here.
Right. Any final questions? Okay, well, thank you very much. This has been fascinating. Appreciate it.
Thank you.
Thank you.